We are live on LinkedIn. Thanks for joining us today. We have a very busy program today. And let me first introduce you my co-host, Gabriel Cian. Hello, Gabriel. How are you?
Hello, Ari. Thanks for inviting me. Great. Thanks for asking. How are you, Ari?
Perfect. It's a great pleasure to have you today because we have a great guest today. So I'm delighted to have you with me. So maybe just before we start, can you just present yourself and what you do? Uh, I know what you do, but it's always
Sure.
good.
For your audience. Okay. So, my name is Gabriel. I am a serial entrepreneur coming from the tech space. I spent 20 years in entrepreneurship, building AI and big data startups before transitioning to longevity and become a longevity enthusiast and activist. So now I'm working on two projects. I'm co-founder of a startup called iCare.ai, helping people live longer and healthier lives. And I also work on a longevity investment club to help longevity startups get funding by investors.
Fantastic. And I remind people that we have a great talk with Gabriel. Just check the website Episteme Entrepreneur. 1 hour long discussion, passionating. So I invite you to take a look. Just I will onboard Michael. Hello, Michael. Thank you very much for being our guest today. It's a great pleasure to have you.
I was going to say the same thing about you too, Ari and Gabriel.
So maybe just a short presentation. It's I'm sorry in advance because you have such a great journey into science. So I would just present you shortly, but of course I would share all the detail of your great parcours journey in the science and technology in the description of the video, then in the replay. So you are PhD in neurobiology, then a medical doctor, and who dedicated his entire career to aging. You are an expert in the cellular senescence and telomere shortening, and also the co-founder of a great startup called Telocyte. I'm right.
Right? Correct. Thank you.
Would you like to correct something or add some point?
No, I think that sums it up in just a sentence or two.
Perfect. Um, we talked about this also before the live. You also had a long career as a professor, as a university professor. So you have trained a lot of doctors also. So it's a great contribution to the medicine. Also, being a teacher is fantastic. So thanks for that. Um, just to announce the plan of this episode. First, we will talk about a little bit about your background and what was your inner fire, that motivate you to become the scientist, the researcher you are today. And then, if you wish, you can offer us a review of what we have to know about the aging landscape, because it's a fascinating world. Do you agree with this plan?
Yes, that sounds good to me, Ari.
Fantastic. Um, I will try to add you on the. Okay, this is better. Michael, when you were a child, did you have already this fascination about life science and biology, or in the background of your parents, did you perform experiment on insects? What maybe most biologists do. Uh, how was it your environment when you were a child?
No, there was no particular biology background. Actually, my mother was living in Paris before the war. She was working for Vogue, lived on the Champs-Élysées.
Hmm.
And prior to World War II, she smuggled out some of the first concentration camp photos back to the United States, which didn't make much difference in the United States. No one paid much attention. And then both she and my father were active during the war in OSS and then afterwards in the CIA, something I
Hmm.
never knew about till after they both died, which probably is appropriate.
Wow. So you didn't have this scientific environment. So how did you uh, wake up, and discover that science and was something that you want to do as a career, as a study? Was it the last day of high school, or is it something that became even later during the university?
I think later, I think in high school I was initially fascinated by mathematics, particle physics, physics in general. and then got off on a tangent in art, history, and anthropology.
Mm.
Ended up in college going into psychology, and in fact, and had a minor in religion. And in fact, I was offered a position in India to study early Buddhist philosophy, and was going to go there for graduate school. But my advisor talked me into doing the graduate record exam, and I got perfect scores on it, which surprised me, and I think him. So he suggested I go to graduate school in the United States. I borrowed 3 applications, sent them in, drove a moving truck out to California, hitchhiked up the coast, got an NSF grant, and went to Stanford in psych.
Psychology, wow. And the higher education system in the United States is always fascinating, because, for instance, in France, in high school we are already specialized in science or literature or economics. You cannot move, you cannot choose even in university. Soft science or hard science, you have to choose your biologist or your physicist or your philosopher or your literature, studying literature, but you cannot jump and discover other discipline. And this is always fascinating me in the production of the elite in the United States, this ability to offer you this possibility to discover other fields during the first year of university, that it's very important to the enlightenment of the brain. So this is great. So you started by studying humanities before joining psychology, that is a little bit more scientific discipline than anthropology and theology even. And how from the psychology you find that, yes, I have to do a research PhD in neurobiology, because it's once again another move, because you have to learn everything in the cell culture, in molecular biology, and so on. And how this opportunity came to you?
One of the questions that you asked me in an email before we started this is particular obstacles or enemies, people you ran into that created problems. And without naming names, there was a person at Stanford I was working under who wanted to put out a paper, and he confessed to me that he'd made up half the data. And I said, could you just leave my name off of that? But he understood what I was saying, which is that I thought this was unethical. And during our honeymoon, he threw me out of, my wife and I eloped after we'd known each other for 7 days. It was an interesting arrangement. But he threw me out of the department. The head of the university tried to put me back in because I had straight A's and a Science Foundation fellowship. And they ended up moving me over into the med school, where I was involved in neurobiology, which was much better because it's really what I was doing. And then as I finished that, I was teaching a lot of the neuroanatomy courses and things like that in the medical school. They suggested I go to medical school. And they asked me whether I'd like to be a professor or a lecturer. Professor meaning, in this case, assistant or associate professor. And I said, I'd love to be a professor at Stanford, but why would I want to be a lecturer? And they said, lecturers get free medical school tuition. I said, I'm a lecturer. Absolutely. So I ended up being paid to go to medical school, went through in 2.5 years, actually, taught some of the courses, enjoyed it thoroughly. But that's where I first became interested in aging. And in a nutshell, the problem I found was that people were saying, when I'd ask about aging and age-related diseases, they'd say, it just happens. And I thought, that's an awfully blasé and simplistic way of looking at the world. And as I think both you and Gabriel know about anything in life, it is always more complicated than we think it is. And when people say it just happens, it right away raised suspicions to me that people weren't thinking carefully. So that's why I got intrigued.
So very rapidly, when you started to your medical studies to become medical doctor, you were initiated to the aging issue as being a disease, right? So did you have a mentor at that point, a gerontologist who have this mindset already, who for him or for her aging was a disease, or was it something that you discovered by yourself?
I think by myself, but it's funny you asked that question, because when I talk to biologists, often they'll say that aging is not a disease, and often doctors will say aging is not a disease, but they treat it like it is one. For example, these are the same biologists and doctors who are getting Botox injections and dyeing their hair and getting facelifts and knee replacement. They act exactly like these things are a disease. So there's a certain amount of hypocrisy, I suppose. From my perspective, it's much like the medieval arguments about the number of angels in the head of a pin. It doesn't have any particular bearing. Whether aging is a disease or isn't, to me, it doesn't matter. Is max nix. It's just ça m'est égal. It doesn't matter, I think. The real question is, what do you do about it, and how do the regulatory bureaus look at it? For example, the FDA looks at it as not a disease and wants you to go after a defined disease. That's fine. I can deal with that. Other people will say it is a disease. That's fine. I can deal with that too. It's just a difference of opinion.
Yes, but at the same time, you interested yourself to cellular senescence and telomere. So this is kind of etiology, of all the age-related disease. So
Yeah, let me come
you said
back to that. In some sense, I don't care what causes aging. To me, it's irrelevant, and I'll come back to that. I don't quite mean that. It's like when new residents in medicine will come to me, and I'll say, "What's your role?" And they'll say, "To diagnose the patient." And I'll say, "No, that's not your role. Your role is to try to make the patient better." Now, the diagnosis may play a critical role in getting that making the patient better. It may be important to find it out, but the patient didn't come in for me to tell them in Latin what I think they've got. They came in to see if I can make them better. And I think the same is true of aging. In some sense, I don't care what causes aging. Interesting conversation over a nice glass of wine. But the real question is, can we do something about the pain, the discomfort, the fear, the tragedies involved in age-related diseases? Now, to do that, I think we need to understand the aging process. But per se, to me, it's just philosophy. Happy to talk about it over a glass of wine or a beer. But the practical issue is, can we do something? So
Absolutely.
That's been my focus for the last 50 years.
Very interesting, because it's remind me, this endless discussion in evolutionary medicine versus practical medicine. When you talk about with the evolutionists, Darwinian evolutionists, about the cause of disease, we can talk a day, an entire day, about what are the cause of a disease, and the practitioner, practitioner doctor want to know to solve the problem, and it's very interesting. Maybe so we passed through your journey quite rapidly, and I have some questions. Feel free to say it's not relevant if you wish. You could have continued as a PhD, into a postdoc and become a biologist, a neurobiologist, being in the lab, or so you say that you have this opportunity to join the medical study, but how was it for you to quit, the research in the lab, or do you have always had a fit in the lab and a fit with the patient's bed? Or how was your journey during the medicine?
I think it was a sort of a blind man and the elephant problem. That is, when it comes to aging, we know an awful lot, but understand very little. For example, even back 50 years ago, we knew about mitochondrial dysfunction, we knew about glycation, we knew about progeria, we knew a lot of little things about the elephant, but we didn't know there was an elephant. Again, the blind man and the elephant, they know there's a tail, there's a trunk, there's a tusk, there's a leg, there's a side, there's an ear, but where's the elephant? And so the frustration I had 50 years ago was that I saw a lot of avenues you could go down, but where was the elephant? How did you put them together? And the result for me was that I decided to go into clinical medicine, which I enjoyed emotionally. Intellectually, my interests remain with regard to age-related diseases, but emotionally, it was very satisfying, the idea that I can try to make people's lives better. And it wasn't until the early 1990s that I began to see the elephant, and we can come back to that. But that's what got me off into clinical medicine rather than research, is that I saw easily a dozen avenues, but they didn't seem to connect. And the question was, were they dead ends? Were they unrelated? Was there something behind them all that unified them? And it was frustrating for me in the 1980s.
It's really fascinating how you got interested into the aging processes way before it became, a really trendy field. I guess it started like in 2009 or so with the Yamanaka factors, and people started understanding that we could eventually rejuvenate cells. But decades before, gerontology was a very unsexy field. So how did you managed to specialize in this field, while there was little to no money, no public funding for that specific research, and not very much interest on the academia.
Yeah, it was a gradual process, and I actually thought it would change remarkably in the mid and late 1990s when it didn't. And you're right, it took a while before it began to become de rigueur. But what was going on was in the early 1990s, I decided, short of anything else, I'd at least write a textbook on the biology of aging. And I went to a conference in Lake Tahoe in California. And I ran into somebody who I thought wrote a better textbook than I could, barking. And two, I ran into the early work on telomeres and telomerase with Cal Harley and Jaron, which was really the first biotech startup for aging. And they actually invited me to fly out to California and see some of the proprietary data and write it up. For me, it was intriguing because the first data I saw on cell senescence and telomeres had three things that fascinated me. One was that the data lined up. Rather than just being a bunch of spots over a graph and sort of a correlation coefficient that was driven through it with a bulldozer, there was nice data that lined up between loss of telomeres and cell senescence, for example. The second was that for me, it began to put together the elephant. I began to see how I could put together all the anatomical pieces of the elephant, construct at least the vertebral bodies, as it were. The third was that Cal Harley, a friend of mine ever since, who just died last year of Alzheimer's, was an interesting guy. He was very, very careful about his research, and many people are not. They simply say, this proves something. Cal, if Cal had been asked, what color is my shirt? He would have said, the front is white. I can't see the back, so I'm not sure. Very careful guy. He would have guessed the back might have been white, but he wouldn't put that in a scientific paper because the data didn't show that. And many researchers are not that honest or careful or anal, frankly, about it. He was very impressive. So they brought me out there, and that prompted me to write the first book on this whole field called Reversing Human Aging, followed by the first two articles in the medical literature in Journal of American Medical Association on prospects for reversing aging and age-related diseases. I also gave the first talk on this ever at the NIH. And the NIH is fascinating. In those days, the auditorium very steeply raked. So people are physically looking down on you, but they were also conceptually looking down on me because the title of my talk in 1996 in April was Reversing Human Aging. And I knew that they were going to assume that nothing could be done about that, as people often do these days. And so I started my talk by saying something that was, I think, radically honest, absolutely honest, the same way Cal Harley would have been. I said, listen, before I start this talk, let me warn you. Anybody who leaves this room in an hour and thinks you can reverse aging is naive. But anyone who leaves this room in an hour and thinks you can't reverse aging is just as naive. The question you should be asking yourself is, where's the data? So let me show you the data. And there's not much of it at those days, but here's what we have. And we were on from there. Now, if anybody ever turned to me and said, when will we reverse human aging? Historically, when is the point? If I said, when did we cure smallpox, you can pick your time. Polio, you might pick 1954. If I said, when do we land on the moon, you can get it to the millisecond. But say that I go forward 100 years and I said, when did we first cure human aging? The honest answer might have to be 1998, when we first showed in a paper in Science that we could reverse aging in human fibroblasts. Quite handily, thank you. So much for mitochondrial dysfunction and Yamanaka factors and everything else. 1998 paper in Science, very turgid paper, hard to understand. I looked at the data, read the paper, took the author out for a beer, Menlo Park, and then went back and looked at his original data and reread the paper, and I get it. But it was fascinating. The second set of papers were not on human cells, but human tissues, done in the year 2000, 2001, some original work on skin, and then also on vascular endothelial cells and on osteocytes and so on. The problem is you're not just a set of cells, you're not just a set of tissues, you're an organism. Mm-hmm. So the question is, can we deliver and alter aging in vivo, in a living patient? And there the answer stood for a while. So that's where we stood now, roughly 25 years ago. And things have slowly changed since that time. And since that time, in many ways, we have learned a great deal more. We know more, but we still understand not very much because people confuse knowledge with understanding.
Fascinating. I will just share the screen to show your books, because they are fantastic. They are fantastic reads. The last one is aging. So you are the editor of this book with many great co-authors. I deeply recommend this book. I read it cover to cover, and it's fantastic review of everything you have to know for aging, tissue, organ after organ, everything you have to know about the aging of every organ. It's a very great book. I really enjoy it, the reading. Of course, it's for I think the target are biologists and physicians specialized in all physicians. I think all physicians can be interested by this book, and biologists interested by aging have to read this, must read this book. The other ones are Cell, Aging, and Human Disease. The Telomeres Revolution. This was a great read. Also, I just finished it last week. I think it's great book, and this one is maybe the older one. Okay, well, let's come back for the presentation of book. And also, I would like to thank the American Press for kindly providing me a version of your book. It was a great pleasure to read it. Maybe we can then. Michael, maybe if you would like to present something about the review on the landscape.
Sure. As I say, I think one of the problems is that we know a lot and understand not very much. It's sort of a problem of knowing the trees but not the forest. And I give you an example of this. Something I noticed back in early years when I was learning geometry and calculus and algebra. I noticed that, for example, if I'd say in the test, what's the formula for the surface area of a sphere? Most people would memorize that.
Hmm.
They didn't know where it came from, but they knew how to memorize it. They knew it, didn't understand it. I found it was, for me, easier simply to understand the calculus and rederive the formula using calculus because I understood it. And so I didn't memorize formulas, but I understood the background process. And I think this is often what we do in medicine. It's what we do often in science. It's certainly what we do with regard to aging. We know an enormous amount. Many people involved in this field know more than I do, but they don't put it together. It's a classic problem of not only knowing the blind man and the elephant, knowing the leg, but knowing the toenail, knowing the left toenail, third toe, the fungus underneath the subungual fungus. They're focused. And they don't see the elephant. They don't understand how it goes together. In aging diseases, a typical example I see is people know the upstream risk factors, for example, Alzheimer's, 2ApoE4 alleles, recurrent head trauma, encephalitides, all sorts of risk factors, microbiome changes, diets, toxins, exposure to radiation, lots of things are upstream risk factors. And we also know a lot of the downstream outcomes, beta amyloid, neurofilament light chains, tau tangles. It goes on and on of things we know about mitochondrial dysfunction. The question is what goes on between. So a couple of years ago, the Alzheimer's Association asked me to help organize the conference in Washington on animal models. And as you know, one of the classic statements in Alzheimer's is that everything works in mice and nothing works in humans, which isn't true because most things don't work in mice either, frankly. And right. Well, of course, the other classic meme, and Gabriel knows this, is that, Alzheimer's is a graveyard of biotech because most things fail. So I was supposed to summarize what was going on in these talks. And I said, I'm not going to summarize what people said, but let me point something out. First, I pointed out that all of the animals involved in the research, mice, rats, cats, dogs, rhesus monkeys, even humans, every one of these species had bitten me, which is unfortunate for my career, but interesting. I like gorillas a lot better, actually, than rhesus monkeys. Much nicer people, much nicer than many human beings, actually. And the adults, the humans that bit me weren't just children either. So anyway, interesting background. But I said the major problem that we've got is, again, we know a lot, but we don't have a model. We know these upstream risk factors, for example, genetic and epigenetic risk factors. We know downstream outcomes, that is biomarkers and hallmarks of, in this case, Alzheimer's. We don't know what goes on in between. What's the black box? What is it that takes us from upstream risk factors to downstream clinical outcomes? That's what we miss. We miss a unified systems model to explain how the system works. Okay. So afterwards, I got a lot of people took me aside and said, yes, yes, absolutely, we agree with you. And one older gentleman took me aside and said, I totally agree with you. Do you have such a model? And I said, I do. I don't see any reason to impose my model on other people, but it's a common problem we have. We don't even think about the elephant. We're thinking about pieces of the elephant. And he talked to me about that model. I asked if I'd publish it. It turned out he was the editor-in-chief of Alzheimer's and Dementia. So he asked me to write it up. I've never had 8 reviewers all give positive reviews of a paper before, and I've never had 1,000 reprint requests. And the editor called it the most important paper they've ever published. From our standpoint, more importantly, it helps us get interest from entrepreneurs and investors, which it did. But the key is still the same thing, which is being able to explain how you go from 2 ApoE4 alleles to increased and earlier risk and more rapid deposition of Alzheimer's, beta amyloid plaque. Why does that happen? So that, I think, has been the major problem with regard to age-related disease and aging in general. People focus on hallmarks and biomarkers. Hallmarks and biomarkers are not aging. They're important things to track. They're useful to measure, but they're not aging. They're outcomes of aging. By analogy, I could say the same with COVID. If I define COVID as a cough, dyspnea, some nausea, high fever, aches and pains, I can go on and on. Those aren't COVID. They are important things to know about, because if the patient has dyspnea and a cough and fever and high white count, I need to know those clinically. But that's not the process. That's not what causes COVID. That's the outcome. That's the biomarker or the hallmark downstream. Mm-hmm. So we'll talk about 9 biomarkers and 11 hallmarks of aging. Important, because again, if I'm going to measure clinical outcome, I need to know them. But they're not the disease. They're not the problem. They're not the underlying unified system involved. They don't talk about what's happening in the middle here. They talk about outcomes, which are not the same.
And this is how the idea of Telocyte emerged, right? Making the link between all these things and
Yes. Originally what happened is Cal Harley got in touch with me because he wanted to get involved with a company in Toronto, Canada, that was wanted to work on, in this case, Alzheimer's and this sort of model. And he would be the CSO, and I was going to be the chief medical officer. And we had a problem with that. I a couple of months ago gave a talk on all of this at Harvard Business School, invited lecture about the biotech and aging process. And they wanted me to talk a little bit about what creates success in an up and coming biotech. And I said, you can get lots of people talking about success, and it seems to be fashion of the day. Fortune magazine seems to have an article about every week on what causes success in a company. And there are thousands. I've got a dozen above me right now, books about how you succeed in a company. I said, I can't talk about that, but I can talk about how to fail. Because the first thing I know about failure is that if you quit, you've guaranteed failure. And the second thing has to do with that original biotech company, which is if you work with people who don't want to be ethical, don't want to work, want to get their egos involved, don't want to follow FDA regulations or EMA regulations or this SEC regulations or the law in general, these are disasters. And I have a number of other things I gave them to talk about, how do you predict failure? How do you ensure or guarantee failure? So for that first company, it was a matter of dealing with people who frankly didn't think they should follow SEC regulations, which is, whether you like them or not, it's a bad idea not to follow them if you're going to start a company. The second, that other comment I made about quitting bears on the current biotech, which we got started 9 years ago and thought people would pick up right away. Back in 1996, I thought people would pick up on the idea of reversing aging pretty quickly. They did not. So it's taken us 9 years, but if we had quit, we would have failed. We haven't quit. We haven't failed. I think we'll succeed.
Fantastic. Gabriel, please.
Yes, I have a question. When I look at Telocyte, you seem to consider that telomere shortening is super important in the aging process, or at least when it comes to the neurodegenerative diseases. And as you have mentioned, there are 11 or 12 now, or even 14 hallmarks of aging, and shortening of telomeres is just one of them. What's your intake on the rest of them, and why do you consider, if you consider the shortening of telomeres like the root cause?
First of all, let me reiterate, I don't care what causes aging. I just want to know what's the optimal point of intervention clinically and financially. The second thing is that some people have said that I think telomeres cause aging. Telomeres no more cause aging than having a laptop caused us to be on this call. these are complicated things. I mean, if I said, what caused this call, what are you asking? And the same thing with regard to aging. You know, I could say it's caused by evolution. It's caused by genes. It's caused by epigenetic changes. It's caused by behavioral changes, medical history. It's caused by a million things cause aging. What do you mean cause? It's a sort of a slippery concept, and it tends to hide our inability to look at things in a complex way. And the same is true of all those biomarkers. Here's an example. The last couple of years, I was chairing a conference on age-related diseases globally. And the last conference, we had 2-3 days of speakers, and some 6-8 of the speakers got up and said mitochondrial dysfunction causes aging and age-related diseases. Well, we've known about that for in excess of 20 years. A friend of mine published on that extensively, Ed Burns, and he has the same concern I have, which is if mitochondrial dysfunction causes aging, what causes mitochondrial dysfunction? You beg question. We had 2 or 3 speakers that got up and essentially said all age-related diseases are caused by immune function failure, that is immunosenescence. Absolutely understood. But what caused the immune failure? And if you said it was caused by glycation, it was caused by Yamanaka factors, the question is, okay, but what caused those changes? Or as some people have said, it's a matter of disruption of DNA data in a sense, the genetic changes. All right, but what caused that? And again, you see the same thing I saw 50 years ago. People wave their hands and they say, it just happens. It's wear and tear, it's entropy, things fall apart, they rust, what do you expect? No, what you've done is you've cheated. You haven't looked back and said, what caused the mitochondrial dysfunction? What caused the immune senescence problem? What caused the glycation? What causes the disruption of genes? And you need to put this in a broader context. And most of us, if asked how old we are, we'd say, well, we're a couple of, several decades. I'm 73. But in fact, all of my cells come from fertilized ovum. That ovum came from my mother. It was already several decades old. It came from her mother, and that was several decades more. And if you track back that line of cells, you'd have to argue that I'm something in excess of 3.5 billion years old in the cellular provenance view. Okay, true. So what you've just said is that the cellular line essentially showed no evidence retrospectively of aging for 3.5 billion years. And then clearly there's an inflection point and it ages over the last 7 decades for me. So what caused that change? Why did that occur? And again, in some sense, I don't care. I just want to know where to intervene. If I look at, let's see, if I check. All right, here's a typical example. I'll throw this up to share. There it is. This is taken from our textbook. Or it would be. Oh, I see. Window. There it is. Okay. Done. It should appear for everybody in a minute. This is from one of the chapters in the recent academic press textbook. And what you're looking at is a series of upstream variables, a series of downstream diseases in this case, and in between there are these altered cellular processes. And there are numerous. You see some of them listed there. Many of them have corresponding definitions and hallmarks and biomarkers that are in the general literature, the lay literature. But the question again is, where can you intervene? And is there a place that I can change this? And I would argue that the most effective, the optimal point of intervention for efficacy is the telomere shortening. And I say that for 2 reasons. One is theoretical reasons, which is that it's a nice handy point that everything seems to go through from upstream to downstream. And that includes mitochondrial dysfunction, inflammatory changes, and so on. We see all of these bear back on changes in telomere erosion, and in fact, generally on cell senescence. The second is a practical reason, which is that, again, we've known now for 26 years that if we reset telomeres, we can reset aging in human cells. We've known for 24 years that we can do that in human tissues. That is, you can, for example, grow old human skin into young human skin on an immunocompromised mouse if you reset telomeres. We've known that we can do this within technical limits in vivo in mice for now a little more than a dozen years. First from a Harvard study and second from the study in Marie Blasco's study in Madrid. So the question is, can we do this with humans? And I don't mean to overplay this because, I'll get rid of that one. Because there are a lot of issues involved in doing this. So I'll pull up some of these from, again, let me pull this one up. There are a lot of things we don't know. So this is an example of from the from the last chapter of our textbook. These are issues that still we don't know about. For example, if telomerase does so well in cells and does so well in tissues and does fine in mice, what's the optimal way to deliver that? There's a lot of concerns about that. And these are open questions. Also, how do we get it in? How do we make it work? How effectively can we make it work in vivo? Because again, in mice, we can reset a lot of things in mice, but how well? How do we optimize transduction from say 1 to 5% up to 90% in vivo in, for example, microglia and neurons? Assuming transduction, do we actually extend telomeres? And then how far do we actually reset senescence in cells? Again, we know we can do it in cells. We know we can do tissues. We know that we can do this in mice, but how well can we do it? And what does this actually mean? And part of that has to do with what I call not only the scientific issues of making this work technically, but they're also what I think of as Humpty Dumpty effects. So for example, if I'm looking at Alzheimer's, if I have a patient who's in terminal stage Alzheimer's, I almost certainly can't get them all the way back. How far back can I get them? If I've got somebody who's in a moderate phase Alzheimer's, where they clearly have cognitive deficits, how far can I get them back? Probably further than I can get back somebody who's in a terminal stage. Likewise, if I'm dealing with an MCI patient, a mild cognitive impairment, I can probably improve that. But how far? And these are a lot of open issues that are very practical. We don't know. So some people have said that, I know more about telomeres and cell aging and age-related disease than anyone, but I don't know nearly enough. I just don't. And those are things we're going to have to find out. There are a lot of open questions that are going to take us to clinical trials. As I used to say, both to the residents and to grad students, having a theory that is consistent with all the data, clinical and laboratory data, and is internally consistent, predictably valid, a nice, elegant theory is excellent. But data wins every time. So what we're looking at is a theory that explains all the clinical data, the research data, the outcomes. It's supported in the lab, it's supported in clinical studies. But does it work when we take it to clinical trials? And the answer is, we'll find out.
Okay, that's absolutely fascinating. So as far as I understand, you consider telomere lengthening or working on this field as the most promising solution. But why hasn't this been more trendy in the last years? Now, the very trendy hypothesis is more cellular reprogramming. There's a lot of money invested into this, and everyone is all the rage today. Why do you think working on the telomeres is not that interesting? Is this only a trend issue, or there are underlying issues with it?
It's a myopic question. I mean, it's a myopic problem. People always tend to look at the superficial part. If I went back 1,000 years and I asked a typical person, not a scientist 1,000 years ago, a typical person, I said, does the sun go around the earth? Does the earth go around the sun? And particularly the Vatican astronomers would have said the sun goes around the earth. Why? Because I see it come up in the east, I see it go down in the west. I'm not moving. I see the sun move. Sun goes around the earth. Well, the problem is it's too narrow a view. That is, you haven't expanded your horizon to look more carefully beyond that. And it took people like Galileo, Copernicus, and a number of others to make that point. But the same thing is true in medicine as well. We tend to look at outcomes without asking where they came from. Again, the classic example is mitochondria. I know mitochondrial dysfunction plays a big role, but why did mitochondria fail? And it turns out that about 98% of the enzymes that are aerobic enzymes that are effective in the mitochondria are derived from telomeres, and their turnover rate slows as I shorten the telomere. And the outcome is you begin to get more degradation of the mitochondrial enzymes within mitochondria. So you have less in the way of ATP and more way of reactive oxygen species, and so on. It doesn't stop there. It gets more complicated. But it's a lot easier to just wave your hands and say mitochondrial dysfunction. So let's try NAD and NAD+, and let's try. It's a simple solution without looking at the details. So it's an uphill battle. It's still a matter of the blind man and the elephant. People focus very carefully on individual pieces, and they don't look beyond that to say, why do these things happen? It's just human nature. We do it all the time.
And in your book, The Telomerase Revolution, I also really enjoyed, to read about, the epopee of Geron, the biotech company founded by the famous gerontologist Michael West, and how this startup or company was important, to highlight this subject of the telomerase. And at the same time, their IP policy was so weird and so, let's say, stupid, that they block any innovation in this field. So it was a fascinating read in your book about this epopee of Giron.
Yeah, it's a classic example. Jaron essentially had three IP platforms, and two of them they initially sold, and they kept one that was least effective. They kept the telomerase inhibitors, but they got rid of the stem cell work and they got rid of the work on telomerase activation. Sold them out, and Mike knew it from the beginning, and it was too bad that involved part of that involved the board of directors of the company. Part of it involved some other people within the company. But really what they did was they lost sight of Michael's goal. Michael is the first person to tell you the same thing I did, which is that, if you think about it, your cells are 3.5 billion years old, your mitochondria approach 2 billion years old. So why do they fail in us? That's an honestly good question. It's not that there aren't answers, but that question needs to be asked, and people don't. They focus on the narrow, they focus on the temporary stuff. I'll give you another crazy example of this. Probably the most influential physician of all time was Avicenna. He wrote a textbook 1,000 years ago, still in print, 750 years later, it's still in use, which tells you that medicine didn't change much for 750 years. But he was remarkable. If you go back to his original textbook, his descriptions of plague are hard to beat. He nailed it right on. He understood exactly what was going on, saw it, could tell you exactly what the prognosis was, identify it diagnostically, but he had no idea that there were invisible organisms like bacteria, viruses, fungi that can cause disease, let alone bubonic and pneumonic and septicemic plague. So he was very good at the details, but he lacked an understanding, not because he wasn't bright, but the time wasn't ready for that. And in some ways, that's what's going on with aging. Most people still assume aging just happens and it's entropy. Entropy plays an enormous role in aging. But the reason we age is not because of entropy, it's because of the failure of maintenance. Maintenance works very well retrospectively for the germ cell lineage for billions of years, but we see that maintenance begin to change as cells undergo senescence. And the usual example is we see a turnover rate of any molecular pool go down. And when that happens, you begin to get accrual of damage as a percentage of damaged molecules go up, whether you're looking at beta amyloid extracellularly around microglia, or whether you're looking at aerobic enzymes, or whether you're looking at lipid molecule turnovers in the membranes of the mitochondria, or for that matter, molecular scavengers for reactive oxygen species or DNA repair enzymes. All of these show a turn down in the rate of turnover. And all of that gets reset with the telomere relengthening. So the question is, does it work? Still, good question.
Gabriel, maybe it's from you. I think the sound of the children coming to your mic.
I'm sorry. I will. I don't hear them, but they're in the house, so I will cut my
You
mic.
said you have
Okay.
the cats, so
No
you
problem.
didn't.
Yeah.
No
Okay.
problem.
So I'll cut my mic. Sorry, and open it only when I need to speak. Sorry about
Perfect.
that.
Maybe we can overview a little bit what is happening right now in clinical trials targeting the different angle of the aging, senescent cells, ROS also. What are the most advanced clinical trials in the field? Do you have some thoughts on that, Michael?
Well, are you talking about just approaches that go after telomeres and cell senescence, or are you talking about things that approach just the downstream biomarkers of it? For example, mitochondrial dysfunction. What are you asking
No,
about?
for example, now we have the molecule that are in clinical trials targeting the senescent cells to kill them. The senolytics. Uh,
Ah,
Other approach. What do you think of these clinical trials? Not on the research side, but now they are more advanced. On assay right now we are talking. So, do we have some data that we can have some hope or delusion?
let's see if I've got. I hoped I'd have that slide there, but apparently I don't. Let's see. Oh, that's chapter 2. Let me see if I can pull up the one I need, which is chapter 13. There they are. Good. Yeah, it's an interesting question, but again, it turns to bear on on a narrow-minded view of what's going on in the world here. Let's see, is this the one on senolytics? Yeah, here it is. Good. Yeah, pull that up for you. This is kind of an overview. Again, this is taken from chapter 13 of the textbook. The problem you have is, the purple line here is natural aging cells. It shows senescent changes over time. And what we'd like to do is the green line and actually reverse aging in human patients. The problem with senolytics is you tend to see this initial improvement and then accelerated pathology. And the reason, I think, is pretty clear when you think about it, which is, let me move that slightly out of the way, but let me give you an example. Say you have a factory, and you have 100 workers in it, and you've got a group of 10 of those workers who are very upset because you've just lowered their wage, okay? That is, essentially the factory is becoming senescent, you've cut back on the wages. Now those 10 workers are very unsatisfied, and they are in fact causing problems for the other workers in the factory, because not only are they unhappy, but they're griping and they're making everybody else unhappy. So you fire them. Well, initially that may be good because they're not griping, complaining, and going on strike. But now the problem is you've got 90 workers and they're trying to do the work of 100 workers. So, you've got some problems. So initially there may be some improvement, but things go downhill even faster. The same is true when you look at, for example, chondrocytes in the knee. What you're doing is removing senescent chondrocytes. That means that the remaining chondrocytes work a little better because they're not having problems with local inflammation, for example, from the senescent cells. But the problem is those remaining cells now need to divide to replace the missing cells, which accelerates their cell senescence.
We lost
so
you.
get this accelerated pathology. So the issue then, I'm going to stop sharing. The issue then is that senolytics are a great thing to invest in now until you get to the FDA phase 2 human trials, and then you're going to start into phase 3. You're going to begin to discover these problems. I think of it as honeymoon effects. Things work well initially, as they should, but in the long run, all you've done is accelerated the underlying problem because you're not treating cell aging. You're treating the outcome of cell aging, that is, cells that are senescent, and not actually reversing the senescence. You're simply removing them and accelerating the problem. In a sense, I think that people who are involved in senolytics aren't thinking deeply about what causes diseases. This reminds me of what we used to run into in the 1980s, where people would have diabetic ketoacidosis, and a lot of physicians would say, well, the thing to do is to remove the acidosis. We'll add sodium bicarb to the IV solution. And what you found is that people got a lot sicker a lot faster because the acidosis wasn't the problem. The problem was what caused the acidosis. The senescent cells in, for example, your knee joint aren't by themselves the problem. They are a problem, but you have to ask what caused that problem and can you reverse the underlying issue?
Fascinating, because I didn't think about that before reading your books. And it was an enlightenment for me, that removing the senescent cell can have, of course, a very short-term good effect, and then the remaining sane cells will have to work more, to sustain the tissue, and it was really very important for me to understand this point. So,
well, there's still a practical issue, which is the question is, but how long is that honeymoon period? So, we can look at that in studies with rodents and get an idea. But let's say you're 60 years old and you've got osteoarthritis and I remove the senescent cells, and 10 years from now it gets a lot worse, but you die in 5 years. And it's a benefit in a sense.
Yeah, absolutely.
How long does it take before you get through the honeymoon period? Does it take a month, 6 months, 10 years, 100 years? And we don't know that. We're looking at rodent data. We're looking at a deeper understanding of how the physiology should work, but we don't know what actually happens until we get to those human trials.
And is there any clinical trial right now trying to rejuvenate with the Yamanaka factor senescent cells in vivo? Or
Well, from my standpoint, the Yamanaka factors are still dealing with, in a sense, downstream issues. Let me use another analogy. Let's say I have a symphony orchestra, and the symphony orchestra has been playing an absolutely beautiful piece of Mozart, and we love it, okay? And now tomorrow we come in and it's playing a John Cage atonal piece, and we don't like it. We'll call that aging, okay? The problem is not that the first violinist has lost her touch. The problem is not that the piano's out of tune. The problem is not that the oboe has broken its reed. The problem is they're playing a different score. And there are two ways to dealing with that. If I've got the orchestra playing a John Cage atonal piece, I could go around to each and every individual Yamanaka factor, each and individual musician, and say, no, play this, play this, play this, play this. And now you still have the problem is that they may not be playing together. A lot more efficient way to deal with that would be to go to the conductor and say, throw out the John Cage, here's the Mozart score, play that again. That's what we do when we reset aging in cells. And again, it resets the Yamanaka factors, it resets the epigenetic changes, it resets the acetylation, it resets the methylation. But how good are we at doing it? Good question. That we don't know. But it's a more efficient way up front, and again, supported by the lab data so far, than it is to go around to each musician or each Yamanaka factor or each epigenetic change or genetic shift than those. So, I think what we need to do is think again in a unified systems view, not in an individual biomarker view.
Fascinating. Fascinating. Gabriel, do you want to?
Yes, I have a question. I'm personally fascinated with neurodegenerative diseases for personal reasons, but also because it's a fascinating field of research. And dealing with the telomere shortening is one of the possible ways of fixing it or improving it. And then there's also some recent research with a neuron injection into the brain, which is very recent. I've heard of it today, where it has shown to improve people's lives, not for Alzheimer's, but more for Parkinson. And then there's another direction, which is really enticing also, which is the brain chip implants, Neuralink style, which have also shown some interesting results in the field. How do you evaluate the different directions, and how could they complete each other to fix this neurodegenerative disease problem?
In some ways, how do I put this? Let me go back to the analogy of bubonic plague or just plague. There were three main kinds of bubonic plague. There's pneumonic plague, bubonic plague, and septicemic plague. And you can define them differently clinically. They are distinct, and the treatments are sort of distinct, but underlying, it's the same process. It's the same organism that caused it. And ultimately the same treatment, whether in immunization or whether antibiotic, same treatment for all three. But they're distinct clinical presentations. The same is true of age-related neurodegenerative diseases. We think of Alzheimer's, Parkinson's, and a host of a dozen others, a whole alphabet soup of them. I pull up the slides and show them. But in some sense, those are artificial. And I'll come back to why they're not. I've got patients with Alzheimer's who have motor deficits, and I've got patients with Parkinson's who have some cognitive deficits. So there's a certain amount of gray zone and overlap. They are real, but many patients aren't classic cases. And the reason is the degenerative process is occurring sometimes in individual locations, like subnuclear, like the substantia nigra in the case of Alzheimer's. I'm sorry, in the case of Parkinson's. But sometimes it's a more widespread phenomena. And ultimately, all of what we're looking at involves cellular senescence and cellular changes. In many cases, again, prompted by, for example, toxic exposures or other things, sometimes vascular issues as well. But there's a commonality here. And all of these cells show senescent changes. Now, if I'm trying to treat Alzheimer's, I'm trying to treat a lot of structures. For example, in the mesial forebrain, the limbic structures, and so on. But they're fairly widespread. If I'm trying to treat the classic example of Parkinson's, I'm trying to treat the substantia nigra, the accumbens, there are relatively narrow neuroanatomical locations. So if what I'm doing is using only, for example, L-DOPA for Parkinson's, it'll have some limited effect because it has an effect on the dopaminergic neurons that are being lost in substantia nigra. But the same can't be said for Alzheimer's drugs. And likewise, if I can replace some of the missing cells in the substantia nigra, or if I can stimulate some of the right cells in the substantia nigra, I could have a positive benefit clinically on the patient. I don't change the underlying disease process. This is like, I'm going to give a gross example. This is like saying, I can replace your coronary artery in a coronary artery bypass graft. I don't change the pathology. I simply take out the bad coronary artery and put in a venous graft in exchange. The pathology remains. It continues. But still, there can be benefits to that, or a knee replacement. The same thing could be true of stem cells in some of the lower brain structures for Parkinson's, or electric stimulation of some of the neurons. But you haven't changed the process. The question is, can we do better than that? And I think the answer is yes. We could, if we really ask ourselves what is going on, not just aging happens, but what's really going on in these cells, then I think we can intervene effectively and reverse the patient. To give an example of this, this is, here I'm looking at the classic process these days. This is from chapter 2. We're looking at CNS. In Alzheimer's and other age-related neurodegenerative diseases, the red line is what happens to these patients. And if you're looking at Alzheimer's, if you're looking at aducanumab and lecanemab, if you look at the actual data, you see this sort of thing. It may even be that it goes back down to the red line again, but there's not a lot of change in the vector, even at best. What we actually would love to do is the green line, which is, can we actually improve function? But that's not what we're doing in almost any of these things. And again, I'll stop that slide, but almost all the approaches we're taking, whether it's stem cells or stimulation or a number of other things, as I say, monoclonal antibodies, it doesn't address the underlying pathology. It is an essential, a clinical Band-Aid approach. That doesn't mean it doesn't have efficacy. It just means it doesn't have the efficacy we'd want.
Okay, your hypothesis is really enticing because it seems to be a sort of a root of all the consequences that happens through aging. But then there is this enzyme called telomerase, and who is lengthening the telomeres. Mm-hmm. Why is it so? What's the limiting factor when it comes to overactivating this enzyme to lengthen the telomeres and fix this problem, or at least show some proof of this concept?
Let me answer that by saying there are two kind of questions you might be asking. One is the downsides, and this is something that Judy Campisi got involved in now 25 years ago in a classic paper about the double-edged sword. And her argument was that telomerase can cause cancer. And the data don't support that. Telomerase is permissive, but not causative. And in fact, if you look carefully at the animal data and the human data for that matter, you can make the argument that, if anything, telomerase may be protective against cancer. But it is complicated. It is by no means a linear relationship. I could pull the slides on that too, and written a couple of papers with a guy at Harvard on this. It is not true that telomerase causes cancer. It is true that we need to think about this carefully. So that's one thing you could ask about. The other issue, though, is how do we get it in? How do we affect this? I'm fairly certain if I can get telomerase in, make it effective, re-lengthen telomeres, I can have good clinical effects. But how well can I do this? For example, the mouse studies originally with Maria Blasco's work suggested that you're only getting about 1% of neurons and 5% of the glial cells, microglia, and yet she had good clinical outcome. But that's a poor transfection rate. So can we do better than that? And furthermore, how do we get it across the blood-brain barrier? How do we make sure that we don't have problems with an AAV causing immunogenicity if you want to repeat the dose in a later time? There are endless clinical issues that bear on this. So it is not as simple as just, sure, we'll put in the telomerase. One, there are concerns, which is why one of our exclusionary criteria is history of cancer. But the other is this issue of how do we actually do it? Not easy. Easy to talk about, not easy technically.
And what is your, because I see on your website that you are choosing the genetic way to activate or to telomerase. What do you think of the chemical molecule, the classical pharmacology approach to?
The problem with telomerase using the protein is, one, it tends to be a very labile molecule in the blood system. Two, you might actually get immunogenicity. 3, how do you make sure it gets through the cells? Turns out that's not easy, although you can address that with lipid nanoparticles. You can address it with viral vectors like AAV or others. But even then, once you get it in, how do you make sure that it gets where it needs to go? So those are tough questions. We're looking at 2 main approaches right now. One is the classic viral vector, for example, in AAV9, which is probably what we won't use, but it's been used in many FDA clinical trials and in the EMA as well. So it's a sort of a known category, even though it has enormous limitations. The other is mRNA.
Just me remind for the audience that the AAV vector are non-integrative DNA vector. So it's a DNA that produces the gene, but the DNA doesn't integrate normally into your chromosome normally.
Yes, I was going to say normally. In a general way, it doesn't, but we know cases where it does, and there's a lot we simply don't know.
And what about the pharmacological approach, with small molecules such as T65, the famous T65 from Giron and others? Maybe there are many others that can work.
The best estimates that we've been able to get, for example, talking to Bill Andrews, is that telomerase activators, that is Th65 specifically, is probably only about 6% as effective as what we'd like to be able to do. We know there are more effective activators, but we also know that they tend to have higher toxicity. And the question is, can we come up with something that's an activator? And there are theoretical reasons why this should be limited. And it has to do with the size of the molecule, what you might call the information or data content. If I have a small molecule, like a telomerase activator, a Th65 as an example, it's a relatively uncomplicated molecule. It's small, and it certainly appears to affect telomerase activation, but it also will affect other things, as any molecule was. For example, penicillin is very effective at intervening or preventing beta-lactam's cell wall synthesis, but it also, as it turns out, causes seizures when I put it on your brain. It's a complicated molecule, has multiple biological effects. And the same is true of telomerase activators. And the question is, can you get one that is only good at activating telomerase and does it 100% without doing other things? And the problem is the molecule is so relatively small, it's hard to pack the correct information content that it does one thing and not the other only. Whereas once you get to DNA molecules, it's a little easier. For example, if I put in a telomerase DNA strand, that's pretty specific. It makes telomerase and it doesn't make much anything else. So you get more bang for your buck in a sense, but it's also a lot harder to get it in and to deal with it. For example, packing it into an AAV vector. In between there, we begin to look at mRNA, which is again fairly specific, has a high information content, it's very specific but how do we get it in? Probably 5 years from now, 10 years from now, we'll be using mRNA exclusively, not just for vaccinations, but for this sort of genetic approach rather than viral vectors. But we're not there yet.
Fascinating. We are reaching the end of this interview. Dear Professor Michael Fossel, it was a great honor to have you, and thanks so much for the clarity of your talk. It was very pleasant to hear you and to understand and to make us understand and shift our way of thinking about this major problem that is aging. Maybe to conclude, would you like to offer some advice to PhD students or to medical students for their career and how they can follow your path and become such a great scientist and doctor like you?
No, not sure I'm great. But I would ask people to focus, on what you know, what you don't know, and realize what assumptions you've made. The other overarching remark I'd like to make is that if I ask what were the most important things we've ever done in medical history, it is not a technical advance, it's a conceptual advance. That is microbe theory. The idea that little invisible things can cause disease made more impact on human health than anything we've ever done. Not a specific antibiotic or robotic surgery, but a concept. I think that we're about to undergo the same sort of revolution with regard to aging. If we can understand it, we're going to do more good for human health than anything that has preceded us, with the sole exception of microbe theory.
Fantastic conclusion. Gabriel, would you like to say some words before we quit?
Yes, it was absolutely fascinating and very interesting to discuss with you, because you have a somewhat different point of view than the mainstream, what we read everywhere. So it was very enlightening for me. And I would have like 10 times more questions, but of course, only 1 hour to ask them. So I'm going to stop here.
Well,
Thank
just
you very
because
much,
it's
Michael.
different doesn't mean it's right, doesn't mean it's wrong. We'll have to see what the data shows.
Thank you so much. Thank you for so much. I will of course publish the replay of this interview on my blog with the blog article with all the link to join Michael and learn more about him and his great books and his great startups, and also the link to connect with Gabriel. Thank you so much for being today with me.
Gabriel, thank you. Ari, thank you.
Thank you, Michael. Thank you, Ari. Have a great day. Bye bye.